Dear students, in today's lecture we will continue what we were talking about in the last lecture, that is bioremediation, and we will be focusing mostly on degradation of contaminants. We will start with metallic contaminant as we talked about in the last lecture, and then we will go on to organic contaminants. So, let us get started. If you remember, in the previous lecture we discussed what is bioremediation, and the most popular definition of bioremediation is degrading organic compounds using biological activities. However, I did mention very clearly many a times that bioremediation is broader than just degradation of contaminants or sequestering them. Okay, so and we talked about metal leaching in the previous class, how we actually use microorganisms and their activity to get more metals out of poor quality ores, and we also talked about acid mine drainage, which is what happens when the metals are leached out and because of sulfate oxidation, and as a result we have highly acidic and highly acidic surface water, groundwater, which have lot of heavy metals in them, and which is a health and environmental problem. Now we will go to uranium bioremediation. In many parts of our country, India, and definitely across the globe, uranium mining holds promises of generating a relatively cleaner and more efficient source of electricity. Now, a problem with uranium is that when we mine the uranium, it gets exposed to oxygen. So, if you remember what we talked about in AMD, that is acid mine drainage, that when the ores get exposed to oxygen, they start getting oxidized, and in case of most metals, or certain metals at least, the oxidized form of the metal is more soluble in water than the reduced form. So, uranium has two valency states that are of concern here. One is the uranium 4 and the other is uranium 6. Alrighty, so the more oxidized one is the more soluble. So, we want to reduce uranium so that it remains sequestered and it does not dissolve in water. And in the sites, this research, most of this research has happened in USA, because there was a time when they were very eager in mining uranium and generating cleaner energy source from there. However, now this research, well, the uranium mining has diminished for different reasons because of change in policy there, but this remains to be a major environmental concern. The presence of uranium in groundwater, presence of uranium in soil, wherever uranium mining has happened across the world, and definitely in India. So, when we mine uranium, it gets oxidized, and the the harmful thing that happens is that the oxidized uranium dissolves in groundwater, because now it is more soluble, and then it is transported over long distances. So, many a times people actually can detect uranium in the drinking water and in the irrigation water, which is a health hazard. Okay, so here is a picture showing how uranium might transfer from one part to another, and this is true for any contaminant actually. Here we have a factory, it is disposing its waste, so it could be mining factory, mining industry that has its spent ore that it is disposing, or it could be a petroleum factory, or it could be any other factory. When they usually keep their waste underground, and if there is any rupture in the underground tank, the water will take the contaminant along with it, and it can be picked up by the wells downstream and by the surface water downstream. So, when it comes to uranium, as I mentioned, it has two forms, uranium 6 and uranium 4, and uranium 6 is the more oxidized form, obviously, because it has lost 6 electrons. So, what we want to do is convert uranium 6 into uranium 4. In presence of oxygen, it becomes uranium 6, which is more soluble and thus more mobile. But if using any technique we can convert uranium 6 to uranium 4, we will have the less soluble. So, we can write insoluble here to contrast. So, comparatively insoluble and immobile. So, this process of converting uranium 6 to uranium 4 to render it less soluble and immobile is uranium sequestration. So, the earlier approaches for doing uranium sequestration involved chemical reactions where people tried to chemically reduce the uranium 6 into uranium 4. However, Mr. Dr. Derek Lovely realized that there are some microorganisms that will convert uranium-6 to uranium-4. And it's very easy for them to do it, even though they do not use the energy when they they should get when they convert uranium-6 to uranium-4. So if you remember, we talked about microbial energetics. If microbes reduce uranium-6 to uranium-4 and they get some energy out of it, then this energy they can use for their sustenance and replication. However, uranium, we do not know if microbes are able to use this energy. We believe that they do not use this energy. That's what evidence we have. Then the question is, why would microbes then want to reduce uranium if they don't get any energy from it? And this is where Dr. Derek Lovely's work is really fascinating, because he realized that he observed that, and he gave evidence in favor of this, that microbes do not metabolize uranium out of their selfish interest, but it is a co-metabolism. It just happens to be so. And why it happens to be so? Because uranium reduction gets coupled with iron reduction. So usually in uranium mine, iron is also present in two forms, ferrous and ferric. So ferric is the more oxidized form and ferrous is the less oxidized form. So the iron reducing bacteria, when they reduce ferric to ferrous, they gain energy from this this process, and then the enzymes and the proteins that they use for carrying out this reduction, just by mere presence of uranium-6 in their vicinity, will also catalyze this reaction. So if you remember, I have talked about this, that metals, heavy metals particularly, microbes cannot bring it inside their cell, inside their body. So these enzymes that iron reducing microbes will use to convert ferric into ferrous, these are extracellular enzyme systems. So these extracellular enzyme systems, just because in their vicinity there is another metal that is oxidized, and it so happens that this metal can also be reduced using the same enzymes, even when this energy is not being captured by microbes. This is how we do uranium sequestration. Now, how can we encourage iron reducing microbes to reduce iron? Now remember, if this is a reduction of iron, which means iron is electron acceptor, so we need an electron donor. If you remember, electron donor plus electron acceptor, you get energy, microbes thrive. So what we do in order to encourage iron reduction is we add an additional electron donor, and the typical one that is amended in the soil or in the water is acetate, because acetate is very easy to digest for iron reducing microbes and most microbes in general. However, acetate does not disappear like glucose does, so the effects of acetate are expected to be long run. Now in this research there were some very other wonderful observations that came into picture, which was that not only do microbes drive the chemistry, so this is biochemistry in sense, not chemistry of biology, this is biochemistry in sense that microbes drive the chemistry and then chemistry affects the microbes. So there is a big relationship between microbes and the chemistry in the soil. Now because this is happening underground, we notice that when microbes affect the chemistry, which is when they reduce the uranium or when they reduce the iron or oxidize the iron, they also not only affect the chemical state of iron and uranium, but they also affect the mineral structure. So they affect the kind of minerals we will find, the kind of crystals these minerals will make underground. So for example, will they make framboidal pyrite, or will they make some other kind of pyrite once iron has been reduced? Now that is why this field, where we look at how microbes are interacting with the ions, the chemicals in the ground, and how they are affecting the structure of earth, this is an entire field in itself, and it's called biogeochemistry. Biogeochemistry. So biology is determining the earth structure, is determining the chemistry, and it's also the other way around. The chemistry affects what kind of microbes will grow and how much and why, and same is true with the geology, because with certain minerals or upon certain minerals, microbes might find it easier to make biofilms and thus easier to survive. This is a very fascinating field right now, and again, for students who are interested in higher studies, I highly encourage you to look up biogeochemistry and good researchers in this field. Alrighty, so here we have a bioremediation strategy for uranium, which is uranium sequestration. We drill a well and we inject acetate. So when we inject acetate, the acetate is consumed by is oxidized. So acetate is electron donor, it's oxidized into carbon dioxide, and that microbe, what it does is it reduces iron and co-reduces uranium. So the microbes that the Dr. Derek Lovely's lab isolated were geobacter, and they belong to this particular species. So here is a clearer picture. We have uranium bioremediation, acetate is being oxidized, the electron is reducing iron and it's also co-reducing uranium, and because of this reduction now uranium is precipitating. Here uranium was not precipitated, but the groundwater is flowing from right to left, and here the uranium is now precipitated because of acetate addition. Alrighty, now let us move on to oil spills. Recently in Chennai there was a major oil spill, and that affected the coast, and we don't even know the expanse of the oil spill. For example, the data about how much oil has spilled, how much area was damaged, what kind of damage has occurred, that data is yet to be arrived in public, yet to be declared on public domain. Now when oil spills happen, it is not just that the environment gets dirty, but it seriously affects the aquatic life, the avian life. So here is a picture on the left of two birds that have been drenched in oil, and they are probably going to die, or they are already, if not already dead. And in fact I encourage you to go and look up the pictures of oil spill in Chennai. And one of the most recently photographed oil spill was Gulf of Mexico oil spill in April 2010. So go ahead and look up the pictures and see the massive damage. It's really beautiful, because if you can see the pictures you can get a second-hand experience on how much damage does it do to aquatic system, to avian system, how much it damages our coast. And you know, many animals such as tortoise use coast to lay their eggs. So when oil spill happens, damages the coast, their young ones die. So we are already in middle of mass extinction, and oil spills are really bad. So we need to have a way of clearing the oil. Now if oil is there in the ocean, how can we remove the oil so it doesn't affect the fishes, the aquatic system, or doesn't affect the birds that feed on these fishes? If the oil is on the coast, we need to know what is the best remediation strategy to clean the coast. And if the oil is in our groundwater, we need to know how to remediate it, how to remove this oil. So let's look here. Let's see now across the globe, many of the major oil wells and oil fields which serve as the major source of oil spills. So either the oil spill will happen at the time of taking it from the ground and sending it to refinery, or it will happen during transportation. So if it happens when we are taking it from the ground, most likely we are taking it from undersea well, and if oil spill happens in the ocean, this is a picture trying to say what its multiple fates might be. It is quite, let's start from the left side, it is quite possible that the oil that has spilled somewhere will finally come and get trapped under the land. So if there is an overhang, for example if there is a solid ice, multi-year ice that is like a permafrost, then the oil will be trapped here, and we will not notice any oil contamination on the top or from the surface, but it is here. It is also quite possible that when the ice keeps growing, so every year when the winter comes, these oil that has been pooled in the bottom of the multi-year ice might get trapped. So we might have ice encasing the oil pocket. It is also possible that and well, depending on the oil, its more soluble components like aromatic components will dissolve, and they will have direct impact on the aquatic system, because most of them are very toxic. And because they are soluble, remember microbes, they have bilipid layer, so they expect that on the both sides of the bilipid layer you have aqueous mixture. So inside aqueous media, cytoplasm, outside also aqueous mixture. So microbes, they like to live in water or in moist places. Now so they cannot enter oil and degrade oil, they can only do it on the surface. So when the water soluble components get dissolved in the water, then microbes can easily come and degrade it. So these usually degrade pretty quickly. The other is quite possible, so in different ways it can get trapped, it can make pools, sheets and pools on the surface. So this was observed in Gulf of Mexico, where when after the oil spill in the Gulf of Mexico, the oil reached the coast of USA, that the oil had either made tar balls or it had made sheets. So it can make sheets. Now sheets are tricky because they do not allow the oxygen to they limit the exposure to oxygen of the oil, because remember aerobic degradation is faster than anaerobic degradation. So the aerobic microbes will be degrading it on the top and anaerobic will be doing it at the bottom. Now this gets tricky because anaerobic will take long time, so these reduce the effective surface area for aerobic degradation. The other thing is the oil that is pooled up on the top can percolate into the groundwater, can percolate into the ground. Okay, the other possibility is that the oil might just form a slick layer on the surface of the water body, and again the soluble components will dissolve. It is also possible that because of the movement it might form missiles, emulsion, and they might go up and down, just might roam around, and then once they are pushed down they might again coalesce with each other, make larger droplets and come back up. It is also possible that they undergo photodegradation, so because of exposure of light, some of the volatile components might evaporate. They might undergo horizontal and vertical diffusion. Some of it, the heavier particles, might actually sediment and then might be degraded and released from sediment. A lot of it gets uptaken by biota, alive beings, aquatic beings, and then this might wash on the shore or might go from one place to another, might disperse, form missiles. So this is the usual fate of oil if it's spilled in near ocean. If it is spilled on ground, then this is how it looks like. You spill the oil here, and if there's groundwater aquifer here, it will make a plume. So it makes two kinds of plume. I think I've talked about it. Well, it makes plume, and because it is not water, it doesn't mix with water, it makes a napple plume. So napple is non-aqueous phase liquid plume, and now depending on its characteristics, in its specific gravity, its motion will be different, and it will eventually be transported in the direction of the groundwater. Alrighty, so when it comes to bioremediation, if these are my tiny oil droplets, and good news for bacteria because these are really small droplets, they have larger surface area. So when it comes to petroleum degradation, one of the limiting feature is that we want the surface area to volume ratio. We want to increase the surface area to volume ratio for the oil droplets, for the oil, as much as we can, because remember, microbes cannot enter the oil and degrade it. They can only eat it on the surface. So you look here, the microbes in this picture are eating, these are tiny microbes and these are oil droplets, and they're eating it only from the surface. So around the rim, some are sitting on the top, but there is no microbe inside the oil droplet. They won't survive, they'll get ruptured, because remember that the outside layer of the microbial cell membrane is water loving. So if it comes in contact with liquid, it will get repelled, it can't enter, but even if you thrust it in there, it will get disrupted, and the middle layer, which is fat loving, will change its direction, and the cell membrane will be disrupted, integrity will be destroyed, bacteria will die. So we know that the surface area to volume ratio should be maximized. So what can I do if I have this slick here, the slick layer here floating in the sea? So when in Gulf of Mexico in 2010, some 7 years ago, when there was a major oil spill, what the BP did then, the company and the people who were responsible for treating it, they injected a lot of the surfactant. Now surfactant is basically a detergent, Corexit A. Now this detergent, what it will do is it will make missiles out of this oil. It does the same principle that you have learned in your high school chemistry and briefly perhaps revisited in 12th class, which is basically it will take the oil, and because the detergent has a hydrophobic end and a hydrophilic end, the hydrophobic end will attach to the oil and the hydrophilic end will attach to the water, will be loving the water, and then it will make missiles, small droplets with detergent around it. Now because it has divided the oil slick into small droplets, and what it has actually increased the surface area to volume ratio for microbes to come and attack. The other beauty is many a times the surfactant itself is very easily biodegradable. So in that case, the microbes who are not very who don't find it energetically very advantageous to eat oil may first eat up the detergent, and then when they are rich and they are good in number and they are hitting the exponential phase of growth, then it's easier for them to attack and eat the oil. Alrighty, and I mentioned in the last class that some important topics in understanding bioremediation or gene regulation metabolism, we have already covered metabolism, metabolic control, aerobic and anaerobic pathways of contaminant degradation and transformation. We will be talking about them today or in the next lecture. And then the enrichment cultures. So we have talked about enrichment cultures, what are different ways of enriching them, so we will not be talking about them. And then bioremediation technology, these are important to know. I have already talked about molecular biological fingerprinting tools, and most commonly used and accepted nowadays are the next generation sequencing technology. Okay, now let us look at some xenobiotics that in which we can apply our bioremediation understanding to degrade them. Now in the last lecture I talked about xenobiotics, and I told you these are compounds that earth has not seen before until man developed them, invented them and produced them. So some of the tricky ones that have really affected our environment are DDT, and I hope you know why DDT is bad. Okay, DDT is used for preventing, for killing the bugs that bite us, like mosquitoes, right? Now the problem with DDT is that it gets into the cycle of the food chain of food cycle of many organisms including humans, and then it affects the integrity of eggshells, and thus many birds have died out. This is really sad. Then we have malathion. This is also used for same reason, for killing mosquitoes, and this organophosphate is quite recalcitrant, doesn't degrade very easily, and it is very toxic for humans and for many other life forms, and it also accumulates in the food cycle. Then this is 2,4-D, it's a herbicide, not again very recalcitrant again. This is atrazine, another pesticide, not good for, it also accumulates in the environment and is not good for life forms. This is monuron. PCBs are a major nuisance because they have destroyed many aquifers, and the water have deteriorated water quality of many aquifers. And this is trichloroethylene, very difficult to degrade most of them. Now we can use our bioremediation techniques, we can use our understanding of microbiology to degrade them. So now these are some more xenobiotics for you to look at, but one thing you should notice that malathion and trichloroethylene don't, but most of them have an aromatic structure, aromatic ring or polyaromatic. These are polyaromatic structures, they have multiple aromatic rings. Now the aromatic rings give the compound, this particular compound, high stability, because once the electrons are in resonance their energy level drops, and now they are very stable, and because they are very stable they are not amenable, it's not easy for them to undergo chemical reactions, and in order to make them, transform them or degrade them, we have to add energy. So we get a very big energy barrier just because of the presence of resonance. Now this is your herbicide 2,4,5-T, and look how degradation of microbial driven degradation of 2,4,5-T herbicide looks like. So 2,4,5-T has acetate here, and then has 3-chlorine, and the first step is that acetate is removed and replaced by OH. This is catalyzed by enzymes, and then it loses 2 of its chlorine, which are replaced by OH. Then it has a dioxygenase enzyme that comes in. What it does is, dioxygenase, as the name suggests, it will add 2 oxygen. So one oxygen is attached here, one oxygen is attached here, and when that happens the ring breaks. So this carbon, instead of attaching to this carbon, now we are attached to oxygen. So we have 2 acetate, the ring has broken. Now this is very important enzyme because this is the enzyme that breaks the ring, dioxygenase. Once the ring is broken, the aromaticity is gone, and now this can be easily degraded. So this is the chlorine is lost and it's easily degraded into succinate and acetate. They are further taken to citric acid cycle. We have talked about citric acid cycle, and then they can be degraded. Okay. So in this way we have degraded 2,4,5-T, a herbicide. So chemically it is going to be very energetically expensive and environmentally harmful to degrade it, but biologically the microbes will use their enzymes and degrade it. So why do microbes bioremediate? Why do they degrade these organic compounds? Well, what do they get from them? They get carbon source. So here you have so many in this herbicide you have so many carbons, and all of them can serve as carbon source. They also get nutrition. They get electron donor or they get electron acceptor depending on the contaminant, and sometimes they get none of the above. They're neither getting energy, nor getting carbon, nor getting electrons, nor giving electrons. So why would they do it then? So if you remember what we talked about in uranium sequestration, uranium is sequestered in a it's co-sequestered, it's co-reduced, because the microbes are trying to reduce iron, and when they reduced iron they get energy from it, so it's advantageous for them. But the enzymes that are reducing iron also happen to reduce uranium, and they're extracellular. So if uranium is sitting in proximity of iron, it will get reduced too, and it will get sequestered. So these are the reasons why microbes will bioremediate. So mostly for their own benefit, and sometimes none of the above. So when they get carbon and nutrient source from the contaminant, this is anabolism. So basically they are using the contaminant as a way to increase their biomass. So carbon will help make them more biomass, nutrient will give them elements required, nutrition required for sustaining their life, and if they are using it as electron donor or acceptor, they are using it for catabolism. Okay, now one of the problems that I think I have talked about this in brief earlier, acclimation. So in acclimation, the microbes, oh yes, I did in the last lecture when we were talking about acid mine drainage. If you remember, I mentioned that the microbes that should be from, let's say, cow manure microbes, that should be reducing sulfate and thus allowing the metal to be precipitating as sulfide, these microbes, when they were put in the field, they were not very happy. They did not do a very good job. In fact, it was as good as them not being there. The reason was they were not acclimated to such low pH and such heavy metal load. So if acclimation is adaptation, giving the microbes, giving an opportunity to get used to the contaminant, because many of these contaminants are xenobiotics, or if they are not xenobiotics, the microbes that can do a good job at degrading them or removing them or transforming them, they are not used to it, they are not familiar with it. So we need to give acclimation time, and once microbes have got their acclimation time, they are used to it, then they can degrade it rapidly. So many chemicals are degraded rapidly after an acclimation period. And sometimes if I want to skip the acclimation step, I can add a second chemical. So if you remember, I talked about how surfactant might actually help microbes degrade the oil, is that first step, it increases surface area to volume ratio by dispersing the oil. Once dispersion has happened, it also serves as a carbon source. So microbes that are not used to the oil can first feast on the surfactant, which is not hydrophobic, and then when they are healthy and they are acclimated to the presence of oil, they can start creating enzymes that will help them eat up the oil. So if we add a second chemical like we added surfactant, it can, or electron donor, it can help, or acceptor, it can help skip the acclimation step or reduce it. Why? How does it work? And why do microbes need acclimation time? First of all, environmental conditions may not be favorable. So microbes that can reduce sulfate, now we are putting them in highly acidic environment with heavy metals present in around them. It's not favorable for them, and they'll die out. And the other reason might be the initial population is very small. So I have like a small vial of microbes, and I have an entire aquifer to treat. It's not going to work. So I would want to grow them, acclimate them to the aquifer conditions, and make sure that they grow in size, big size. And if I put my small vial microbes directly into the aquifer, because the initial population is very small, I may not see substantial results until they reach a population threshold where the activity is substantial enough for me to note. It's also possible that in the environment there are some toxins present. For example, heavy metals in acid mine drainage. So the heavy metals might have toxic effects on microbes and not allow the microbes to do their job. It's also possible that there's some protozoa present and they're eating the bacteria, and microbes are not able to do their job. So and then it's also possible that new genotypes need to appear. Now this is very important. For example, in case of oil spill, let's say there is a lake that has never been exposed, microbes that have not been exposed to oil, but now a tanker comes and it disposes some oil in it. Yeah? So now the microbes there do not know the oil. They need to have evolve into new genotypes that can actually degrade the oil and tolerate the oil. So we need to wait for the new genotypes to appear. So if someone asks, well, there are microbes in the lake and there's oil in the lake, why are microbes not eating the oil? There might be many reasons. Maybe maybe that the microbes that can eat the oil are in small numbers, so they need to grow up enough before we see any substantial degradation of oil. Or it may be possible the oil has some toxins in it, like aromatic compounds that are toxic to microbes, so that's why they're not able to eat it. It's also possible that these microbial community in the lake are waiting for new genotypes to appear, and these new genotypes will be able to degrade the oil. And the other is the energy cost of switching the substrates. So initially the microbes in the lake were eating, other kind of organic matter, natural organic matter, but now they have to switch to eating oil. So when they make this switch, then they have to, there's an energy cost to it. So that's the other reason why there's an acclimation. Their switching is slow. The other thing is enzyme induction and lag phase. So just converting making the enzyme activity substantial enough, and the microbes that can eat oil reach a good phase enough, they have to undergo the lag phase, the exponential phase, and then we will see substantial microbes. So all these are reasons for acclimation, why we need acclimation. Now I have been talking about this, that if a microbe aromatic compounds are very difficult to degrade because the energy level is very low. So let's say this is our substrate A and B, this is what we want to degrade, and these are the products C and D that they will make. And there is a good amount of energy to be made from this reaction that microbes can benefit from. However, there is often an energy barrier. So the question here is, why, if we know that degradation can happen, for example we know 2,4,5-T herbicide can be degraded and microbes are already doing it, why do we want to use microbes? Why don't we use some chemical reagent and then, or we change the chemistry such a way that we cause a degradation? Well, the reason is usually there is a chemical barrier, there is an energy barrier, and this is your energy barrier here, this is the energy barrier. Now if microbes, they use enzymes to jump the energy barriers. Without enzyme, this is your energy barrier, so you require more energy, you require more heat, more electricity, or more whatever for this energy barrier to be crossed and products to be formed. But these microbes, very clever little ones, they make enzymes that reduce the energy barrier, so it's easier for microbes to do it in the ambient condition, to carry out the same reaction in ambient condition. Alrighty, now we'll go through oxidation, reduction of different kinds of contaminants in the next lecture. So briefly I'll just go through the hydrocarbon oxidation, and the next lecture we'll take over other contaminants and other kinds of degradation process. So let's say you have an octane, it's a 8-carbon hydrocarbon. Now the first step would be oxygenation. So monooxygenase enzyme will catalyze this. So basically one hydrogen at the tail end would be replaced by OH. So you are adding one O. So one O, so you need monooxygenase enzyme, and then when you do that, you will release your H, and when you release the H, now you are releasing NADH, which is electron rich, energy rich compound. Now when you have made an octanol, which is an which is the oxidized form of an octane, it has here carbon CO, and then it will undergo dehydrogenation, so it will lose another hydrogen, this one, and now we have an octanoic acid. So here another oxygen will come and attach. So now you have made acid. Now once you have made acid, these things are very easy to break, because so you can how they'll break is that this will break down as acetyl CoA, and this CoA will undergo degradation, and then you will need an energy input, and then you will do beta oxidation of acetyl CoA, and finally you will have 8 carbon dioxide and 32 H, and this will be respired. So this is how hydrocarbon degrades. So dear students, this is all for this lecture. In the next lecture we will take up the oxidation, reduction, basically degradation and transformation of other organic compounds. Thank you so much.